Have you ever sat down to a nice seafood dinner and had that little thought in the back of your mind? Is this actually, safe? Well, today, we're going to find out. We are going to become scientific detectives, following the clues with a real team of researchers to see exactly how they uncover hidden dangers in our food.
So here's the deal. We all know seafood is great for us. It's loaded with proteins and all sorts of good stuff. But the oceans where they live can also be polluted. This quote from the actual research paper really hits the nail on the head. It's this constant balance between amazing nutrition and potential risk.
Which, leads us right to the big mystery here. How do we really know if what's on our plate is safe? I mean, this isn't something you can just look at and know, right? You can't see it, you can't smell it. To answer this question, you need a serious scientific process, kind of like a forensic investigation of our food.
Okay, so here's how we're going to break this whole case down. First, we'll look at the contamination question itself. Then we'll see how scientists collect the evidence, what goes on inside the lab, how they decode all that data, and finally, the big one, how they assess the health risk. Let's dive in.
All right, every good detective story starts with figuring out what you're even looking for. And in our case, the culprits are these invisible pollutants. And our key witness is none other than the spiny lobster.
So, our main suspects? Heavy metals. And listen, these aren't your run-of-the-mill pollutants that just wash away. They're persistent, which means they hang around in the environment for a long time. They build up in the bodies of marine life, that's called bioaccumulation, and they get more and more concentrated as they go up the food chain. And that's exactly why lobsters are the star witness here. They live on the ocean floor, right in the muck where these metals end up settling, which makes them perfect little living records of pollution.
Okay, so we've got our suspects and we've got our key witness. Now, the investigation moves from the what to the where. It's time to hit the field. This is where the scientists have to go out and gather the actual physical evidence.
And this is our crime scene, so to speak. The researchers went to these 3 spots right here along the coast of Bangladesh to collect their spiny lobster samples, getting them straight from the local fishermen.
But what's really wild is how incredibly careful they have to be. I mean, this isn't just grabbing a few lobsters and tossing them in a bucket. Every single step is meticulously planned to protect the evidence. They wash them with special deionized water, so they're only measuring what's inside the lobster, not on it. Then, each one goes into its own airtight bag and straight into an icebox. No chance for contamination.
Okay, evidence secured. Now we leave the coast behind and head into the lab. This is where the real high-tech forensics get started, where all those invisible clues finally get brought into the light. So before you can test anything, you got to prep the evidence. The scientists dissect the lobster into different parts, the tail meat we eat, the shell, the legs, and so on. Then they homogenize it. That's just a fancy word for blending it into a consistent paste, and then they use powerful acid to literally dissolve it. It's a process that basically strips away everything but the metals they're hunting for. And this is the secret weapon, atomic absorption spectroscopy, or AAS. The easiest way to think about it is like a super specific metal detector. The liquid sample from the last step gets run through this machine, and it's so sensitive it can tell you not only if a metal like lead is there, but exactly how much of it is in that sample, down to ridiculously small amounts.
Okay, but how do you know your super fancy metal detector is actually accurate? I mean, how can you trust the results? Well, they test it. They run a test on a standard sample where they already know the answer. That's the certified value column. And just look at these numbers. The results, the measured value, are almost a perfect match. That tells us their method is spot on, and we can absolutely trust the data.
All right, so now we've got a ton of data. But, a giant spreadsheet of numbers doesn't really tell you a story, right? The next step is to actually decode it all, to find the patterns hiding in the clues. So, for example, just take a look at this chart. It shows the amount of lead found in one type of lobster. You don't have to be a scientist to see the pattern here, right? The lowest amount is in the abdomen, the tail meat, and the highest amount is way up in the carapace, the hard outer shell. So, what's that all about? What does it mean? Well, it looks like the lobster is actually trying to protect itself. It's pushing the toxic lead out into its shell and appendages, almost like it's locking the poison away to keep its important muscles and organs safe. It's a pretty cool survival strategy.
Now, imagine you're tracking not just lead, but a whole bunch of different metals across several kinds of lobsters. It gets crazy complicated fast. So they use this amazing statistical tool called principal component analysis. Think of it like a master data detective that sifts through all the evidence at once and finds connections, pointing out which metals tend to show up together.
And this is what that analysis spits out. Now, don't get bogged down by the chart's details. The important thing is to look at the clusters. When you see metals like cadmium and copper bunched up together like this, it's a huge clue for the scientists. It screams, hey, we probably came from the same place, like runoff from a factory or a farm.
So we've found the evidence, we've even figured out where it might be coming from, but now we get to the question that matters most to all of us. What does this actually mean for the people eating these lobsters? It's time for the verdict.
The first step is to figure out something called the estimated daily intake, or EDI. It's actually pretty simple. You take the amount of metal they found in the lobster meat and figure out how much a person would get if they ate an average portion. Basically, what's your daily dose?
Okay, next up is the Target Hazard Quotient, or THQ. And this is where we find out if that daily dose is a problem. It compares the amount you're eating to the official safe limit. All you really need to remember is this. If the THQ number is over 1, that's a red flag. It's a warning sign that the exposure could be a problem over time.
But let's be real. In the real world, you're never dealing with just one contaminant, are you? And that's where the hazard index comes in. It's really simple. You just add up all the individual red flag scores for all the different metals. It gives you the big picture, the total risk from everything combined. And just like before, if that final number is over 1, it's a concern.
And then they got the results. And this one is a real shocker. For just one of the metals, arsenic, the target hazard quotient was over 5 in almost every single lobster they tested. Now remember, the safety line is 1. This was more than 5 times that limit. So, what was the final verdict from our investigation? When the scientists added it all up, the total hazard index, the combined risk from all the metals, was over 1 for every single lobster species they looked at. The conclusion was unavoidable. Eating these lobsters regularly could pose a significant hazard to public health.
So this entire journey, from a boat in Bangladesh all the way to a complex data plot, it really shows you how science works behind the scenes, like a silent guardian for our food. And it kind of makes you wonder, doesn't it? What other invisible stories are out there in our own backyards, just waiting for a detective to uncover them?